So we heard a lot about different approaches to recovery. There are medications, there's physical therapy. The other approach that we're taking is to try to actually regenerate the damaged spinal cord using stem cells. And I'm going to emphasize the regenerative aspect of this stem cell approach because there's still a lot of confusion out there about what kind of stem cell trials there are. And in between each section, you'll meet another child of mine. He was born in June. His name is Josh. This is the first time in the little saucer thing. And so what I'd like to cover today is to discuss why transverse myelitis is a good proof of concept model. Dr. Greenberg did an excellent job of this. I'm basically going to emphasize a few of his points. I'll introduce the types of stem cells that we use that may be useful in TM and NMO. I'll show you some of our preclinical animal studies. Animal models are critical to moving towards a human trial. The human trial is in preparation in parallel with these animal studies, so I'll tell you what that will look like. And then some futuristic ideas that we've been working on as well. So anatomically, with transverse myelitis, we know what we're dealing with. There's damage in the spinal cord. This is an MRI cut through the middle of your body. Here's your chin, here's the back of your neck, and the arrow is pointing to the myelitis lesion. The white part here within the gray column is the lesion. In many cases, it could be permanent damage. There could be permanent damage. It can be small and focal like that, or it could be a long lesion like this that could extend over multiple regions. But anatomically, when we think about stem cell trials, this is what we're trying to heal. You can also cut the neck off, look down at the spinal cord, and get a sense of whether it's right or left. And when we deal with how we're going to get the stem cells in, if we're going to do this as a surgical approach, we'll want to know which side of the spinal cord to inject. So MRI is very useful for this. But again, this is to emphasize that what we're trying to heal here, what we're trying to improve, is the anatomy and the function. And this is the anatomical aspect of TM. So TM as a proof of concept model. Why are we looking at transverse myelitis as a model for stem cell regenerative therapy? The reason is kind of the same reasoning as the Ampera trial. There's only one lesion in many of you. If you have NMO, you may have more than one lesion. But if you have TM, idiopathic TM, there's one lesion. All of your symptoms come from that one lesion. So if we could fix that one lesion, we may be able to significantly improve the outcome. TM can be both demyelinating and it could be destructive. We have stem cells that prefer to make myelin. We have some stem cells that prefer to make neurons and neuronal bridges and circuits. So depending on what kind of TM you have, we may have the right stem cell for it. TM patients are generally otherwise pretty healthy individuals. They have significant disability from the one TM, but other than that, they can usually undergo surgery if necessary for the procedure and can otherwise go through the physical therapy that would be necessary and so on. With TM, as opposed to NMO, there's no recurrence, so there's a clean outcome measure. So if you get your stem cells in October, you're not going to have another relapse in January that could confound the outcome. So for TM, you have a clean outcome. For NMO, you don't necessarily, because you could have a recurrence. On the other hand, if you have NMO, you're already on a drug to suppress the immune system. So if you have NMO and you may be on CellCept, for example, that's a transplant drug. Patients who get stem cells, that's a transplant cell, you may go on CellCept. So NMO patients are already on it. TM patients may have to start it. The spinal cord is a relatively easy access tissue. We know how to access it. It's still surgery. You still have to undergo anesthesia, get a little bone removed for the injection, but it's still relatively accessible. And based on the MRI, we would know where to put the needle in. And then, as Dr. Greenberg mentioned, TM and NMO are both orphan diseases. This makes it profitable for companies to pursue a treatment trial for these rare diseases. The clinical course of TM is very important in considering when the best time to transplant is. This is, so Dr. Greenberg mentioned the third rule. This is another one of those rules that we think we know a lot about, but really we need much better data. We know that some of the recovery occurs over the first 6 months. We think that's most of the biological healing that takes place. And a little more after that, for the next 2 years as you regrow circuits and use and learn new ones. So we think that the best time to transplant stem cells would be sometime after you've gone through all your biological healing, but sometime before all of that healing is done so that you can optimize the healing process with the stem cell transplant. This isn't fixed in stone. This is really just kind of our initial thinking about when the best time to transplant stem cells. Does this mean that if you've had transverse myelitis for 5 years, you wouldn't benefit from that? That's not what we're saying. What we're saying is for a trial where we really want to see the best results so that we can be confident the stem cells are doing something, we want to try to see the best outcome. And we think the best outcome would be in this time period. Here are the challenges. This is the FDA's biggest concern, is that we're going to cause harm. So many of our TM patients have quite a bit of function left. If they had a thoracic lesion and their arms are fine, the FDA's worried we're going to harm their arms. So we're going to go in, put some stem cells in there, in the spinal cord, and they're going to migrate up to the upper part of your thoracic, to your cervical spine, and cause weakness in your arms and just make you a lot worse. We may trigger another TM. These are foreign substances to your body, and your immune system will try to react against them. So that's something to be concerned about. Can these stem cells form tumors? That was the concern back in the early 2000s. These stem cells are going to replicate, reproduce, and form big tumors, and they're going to be harmful. And there are case reports of kids getting transplants in Russia and other places where these stem cells did in fact form tumors. So this is not just hypothetical. So this is what we're primarily concerned about. It's causing no further harm. We talked about accessing the lesion. That's still a challenge. For patients with TM, proof of concept, one lesion, stick a needle in that one lesion, try to regenerate that one lesion. For everybody else, for NMO patients, MS patients, it's more than one lesion, and there's a challenge in trying to access every single one of those lesions. I'll talk about a futuristic idea we have to do that. You have intact immune systems, and your immune systems are going to try to reject these stem cells. These stem cells come from another human being, and if you put another human being's tissue in yours, your immune system is going to react against it. We want to try to prevent that. The spinal cord is a little bit immune privileged. So if you put something in the spinal cord, the immune system may not notice it, but there are tricks to getting the immune system to accept a transplant. And of course, we're going to use all of our experience in solid organ transplant in moving forward with a stem cell transplant in TM. Then we have to get this is the hardest part actually. We have to get the stem cells to do what we want them to do. And you can't just tell a stem cell what to do, and it has a very complex task. It has to go to the lesion, it has to recognize what's wrong, and then it has to respond correctly and try to fix the problem. Now, developmentally, when you're a baby and you're a fetus and you're developing these cells, they sort of have a program. They have an automatic program. So we have that in our favor. It's not like we're starting from scratch. These cells are designed to make myelin or make neurons or make bridges or circuits or whatever we want them to do. That's what they're designed to do developmentally. So it's not like we're starting from scratch. And then ultimately, we want to see an outcome. So it's not just enough that we make myelin. That myelin has to be beneficial. Okay, transitioning. So Josh's two sisters, that's Megan and Allison. This was at the pumpkin farm. Okay. So you may have read quite a few newspaper articles about stem cell transplants. And I think that you do because I get lots of emails about them. This is the typical article you may read, stem cell transplants help MS victims. and you call and you say, I want those stem cells. There's something very important to understand about the current stem cells that are being used for MS and other studies. Most of those trials are not regenerative in their design. They're not trying to regenerate the spinal cord. They have other goals. There are many different types of stem cells, and they all do different things. For these MS trials, they're using what are called mesenchymal stem cells. Mesenchymal stem cells come from the bone marrow. They basically include every kind of cell type that does not eventually form a blood cell. So your bone marrow contains blood cells and mesenchymal cells. These mesenchymal cells can easily form bone. These are bone cells, fat cells, and cartilage cells. And they can be useful in that regard. Horses are getting transplants with these cells for regenerating cartilage in the knees, and it works. And there are companies out there, veterinarian companies, and this is used on an ongoing basis. So mesenchymal stem cells are useful in many aspects of disease. And in MS, they're being tested not on regenerating the spinal cord or the brain, but on their beneficial effect on the immune system. MS patients have relapsing remitting disease. They have an immune problem. And these mesenchymal stem cells have a calming effect on their immune system. Now, if you have TM and you had an event 10 years ago, mesenchymal stem cells are not going to help you because your immune system is not activated, it's not stressed out or upset, and those mesenchymal stem cells won't do anything to your immune system. We know that if you put these mesenchymal stem cells near the spinal cord or the brain, they won't do anything. Actually, they will do something. They'll do what they do outside the spinal cord and the brain, which is calming the immune system. But they don't assist in the regenerative process. So what I'm talking about are so-called regenerative stem cells. And I list them here. You want to make neurons, you want to make astrocytes, or you want to make the myelin-producing cells. Astrocytes are supportive cells, neurons are the cells that help you move, and oligodendrocytes are the cells that make myelin. This is ultimately, a combination of these is ultimately what you need in your spinal cord to regenerate. If you try to transplant these into your spinal cord, they will likely die. They're too mature, they're fragile, they can't be moved around, they're probably already connected to something, so if you slash these connections, they're not going to be happy cells. You really can't grow these up in a dish and transplant them like this. What you have to do is go a little bit earlier. And in cellular biology, when we talk about earlier, we talk about stem cells. So anything from here up is a stem cell because it still has a process to go to get to the mature cell. Going backwards, these cells here are called progenitor cells. They just have one or two more steps to get to their final cell of interest. A progenitor cell that ultimately forms a neuron cannot go on and form a skin cell or a liver cell or something else. It's fated to become a neuron, but it can still divide, and it's small, and it's less fragile, and you can grow them up, and you can feed them all kinds of things. You can even genetically manipulate them to do things that you want them to do. So these progenitor cells are valuable in the sense that you can transplant them. They'll migrate to different parts of the nervous system, and then the trick is to get them to form their cell, to go on and differentiate into the cell type of interest. Going farther back to the cell type that can form all three of these, this cell type is called a neural stem cell. This is the most popular type of regenerative stem cell, a neural stem cell. And it comes from many different sources. You can get them from aborted fetuses. You can get them from adult tissue. This is the hottest area of research now for which the Nobel Prize was awarded last year. You can get them from embryonic stem cells, a little bit more politically controversial, but still, we'll talk a little bit about that. So any source can create a stem cell called a neural stem cell. And then beyond that, you can create these progenitor cells that are very easy to manipulate and transplant. Now, when I started off at Johns Hopkins, I worked in Doug Kerr's lab, and we played around with a lot of different stem cells, and we generated our own. We got them from different tissues, and we played around with them and genetically modified them and did all kinds of things to them. But ultimately, when it came time to move to a human trial, we recognized that the effort and the money and everything else that Dr. Greenberg described in getting a cell from the lab to a patient would be too expensive and too time-consuming for us to do academically. So we decided to collaborate because there were companies out there with the capital and the interest in getting a stem cell to human trial. The first one that made the news is a company called Geron, based out of California, submitted 21,000 pages of safety data to the FDA for this cell type called an OPC, oligodendrocyte precursor cell. So this is a precursor cell that ultimately can only develop into oligodendrocytes to produce myelin. And they got the approval by the FDA to move forward with a human trial in spinal cord injury. This is traumatic injury. And it launched in January 2010. They recruited 6 patients. And then, unfortunately, they ran out of money. It costs $65 million a year to maintain this program. And this was after the recession, and their stock price kept falling, and they just decided, we can't afford this. They ended up selling it off to another startup company, which hasn't developed it yet. But back in 2010, when this was the hottest thing around, we called them up and we said, hey, there's a disease called TM. And I went through this whole presentation with them, and they said, wow, what a great proof of concept model. Here's our cells. Test it in your animal models, see if it works. So we threw their cells into a type of mouse that doesn't make any of its own myelin. These mice only live for about 4 months. They have seizures from birth, and then they eventually die because they have no myelin. And it's the ideal model to put a stem cell in just to see if it could make myelin. So the idea was put the Geron stem cells in here labeled in green. Their stem cells are green. We injected it into the brain, and then we waited. Just see what happens. Do they form myelin? Do they respond to the environment and form myelin? Soon after they're injected, they started tracking along neuronal axons. They started migrating. We inject them in the back of the brain, and they started migrating towards the front of the brain, and eventually made it into the optic nerve, for example, which is a pretty impressive feat for 2 months later to find stem cells all the way in the optic nerve. So this was pretty exciting. Inject stem cells in the back of the brain, and they just start migrating around, looking for their final resting place. And then we hope to find out what they do when they get to their final resting place. And unfortunately, they didn't do anything. They just hung around. They went all the way down the spinal cord, for example, and then didn't do anything. So they just migrated. The hope was that they would form myelin. And we thought, well, maybe the environmental condition is not just right. Maybe they need to be tweaked a little bit. We went back to Geron and we said, here's our idea. And they said, great, we just ran out of money. But there was another company that was launching. They were launching their own trial, a human trial using stem cells. And this disease was Lou Gehrig's disease, or ALS. And they used a neural stem cell called the 566 RSC. We made the same pitch to them. We said, transverse myelitis, proof of concept model. They said, wow, this is great. Here's our stem cells. So we put them in that same mouse model. And again, they started migrating up these tracks. For whatever reason, their stem cells here, this time in red, really enjoyed migrating all the way up these tracks. But similar to the Geron stem cells, they also didn't do anything. They ended up in all kinds of places around the central nervous system and just didn't do anything. So I called the company and I said, your stem cells don't do anything. Here's some ideas about how to stimulate them to produce oligodendrocytes and make myelin. And they said, you know what? We kind of already done that. And we have another cell line that's not in a human trial right now, so we don't have all the safety data to the FDA. But why don't you throw them in your animal model and see what they do? And indeed, they make a ton of myelin. This is a myelin-producing cell. There's the cell body in the middle, and it sends out all these projections. and they wrap around axons in this mouse, and they produce tons and tons and tons of myelin. Here's a myelin track here. This mouse doesn't make any of its own myelin, so anything here in yellow is being produced by stem cells. And frame after frame after frame, we found tons of myelin. And I have this one nice depiction that I sent the company where I attached together 17 microscope frames of one really, really long axonal projection going from one side of the brain to the other. So they're clearly making myelin. And some of these mice are actually living longer than the 4 months they're expected to live. Here's another example. The stem cells are in green, and all the myelin that they're making is in red. So it's exciting that these stem cells are producing myelin. Now, unfortunately, this type of stem cell is not the one that has undergone all the safety approvals by the FDA. So we have a little problem on our hands. We have one stem cell that is FDA approved for safety to move forward to a human trial. And then we have another cell type that makes a lot of myelin that hasn't gone through that process yet. So when we think about a translation to a human trial, we have to think about what the FDA would approve and what would they consider safe. And we also have to consider what would be effective. So safe would be the first line of cells. It's undergone the rigorous process. It's safe in ALS patients. 16 ALS patients have been transplanted, no tumors, nothing really abnormal. But we don't think that they'll form a lot of myelin. On the other hand, we have another cell type that seems to be more effective, but we'd really like to try to transplant into a human. And it may eventually be a combination of these things, or we may eventually try both separately. So the next step is to figure out if making myelin is helpful in these mice. Is adding myelin, for example, in a model of TM, do these stem cells actually benefit the animal? This is the type of mouse study we use. It's called EAE, experimental autoimmune encephalomyelitis. These mice get sick on a scale of 0 to actually higher, 0 to 5, where 0 is normal. 0.5, their tails become limp. Around 1.5, their hind limbs are paralyzed. 2.5, their forelimbs, and so on. And it's an ascending paralysis that's very similar to TM and to NMO. And what happens with these mice is after you make them sick, they eventually become stably disabled. This kind of sounds familiar to a lot of TM patients where they have one attack, one event, and then they remain stably disabled. And there are ways to manipulate this model to make them sicker or better. One of those in a rat model is really unique because it's almost entirely demyelinating. So these rats do not lose their axons. These white arrows are pointing to axons that are perfectly normal. They're intact, they're healthy, they're happy, but they have no myelin around them, and so they don't function very well. And so into this rat model, where we think the axons are ready to accept stem cells to produce myelin, we at first, this again was with the Geron cells, we threw in the Geron cells, and they did nothing. We implanted the cells, and there was absolutely no improvement in outcome, and we predicted that based on the fact that they don't make myelin. So our next step is to do the same experiment with the neural stem cells, the neuro stem cells, and our surgery is scheduled for November 26 now. So again, next symposium, I'll have more data. Here's this next transition. This is Megan, their lollipop with her popsicle. So in parallel to these animal studies are going to inform the human trial, but we're moving forward with a proposed human trial based on what we think and what we know about TM. And this is what it's going to look like. The proposal is already drafted. We're working on the consent form, and it's going into the FDA with the following characteristics. It's going to be transverse myelitis, single lesion. Again, we'd love to do NMO, maybe inject optic nerves one day. We'd like to include ADEM, maybe do brain injections one day. But first, we just have to demonstrate that these stem cells work. So we picked TM. Our collaborators are going to be at the NIH. They're interested. We met with them, the National Institutes of Neurologic Disease and Stroke, and a new NIH center called National Center for Regenerative Medicine. It's basically the new stem cell institute at the NIH. They're interested. And if we can get approval to do this at the NIH Clinical Center in Bethesda, the government would subsidize the surgery, which would reduce the price from $120,000 per patient to $30,000 a patient, which would be very nice. So if we can get that approved, it makes the study a lot more affordable. We plan to recruit 6 patients. The first 3 patients will be Asia A patients who have no motor function, no sensory function, and no bowel or bladder function below the level of their injury. This is designed to assure the FDA that we're not making things worse. So we plan to inject the stem cells into the lesions of these spinal cord patients, and we honestly don't expect them to make a huge recovery. We think there's a lot of damage. There's a lot of glial scarring. We don't know what these stem cells are going to do. This is really a safety study to show the FDA they don't form tumors. This is a different population from ALS. They don't form tumors. They don't do anything really bad. And they don't trigger another TM event. That's what these patients are for. They're the real guinea pigs. Then we're going to try to recruit 3 patients with the Asia C score. Asia C is where you have some motor function. And this is really the proof of concept study. This is where we want to see if adding stem cells to the lesion to try to regenerate the myelin can improve the function that they have. So this would be someone in a wheelchair who can move their legs getting out of a wheelchair. That's the model. It's invasive. It's going to be a direct surgical injection. We haven't decided yet if we'll take cervical lesions. The thought is, first and foremost, we can do the least damage by doing thoracic cord injections. Cervical lesions would require a little bit more planning and thinking and maybe a little bit of FDA shrugging, but we haven't decided yet if we're going to try to recruit cervical patients. I think it depends a little bit on what kind of patients we were able to recruit at the beginning. So here's our futuristic idea for patients who have more than one lesion or who don't like the idea of having a neurosurgeon open their back. The idea here is try to deliver the stem cells through a blood vessel rather than through surgery. Now, for those of you who have undergone a catheterization, maybe cardiac catheterization for heart disease, or an angiogram for spinal cord disease, or something like that, you'll know what this is about. This is about putting a catheter into an artery and then releasing a few stem cells into the artery and letting the artery deliver those stem cells to the right location. The way we designed this is we genetically modified the stem cells to express a sticky protein. But it's only sticky if the CNS tissue, if the tissue in the brain or the spinal cord is inflamed. If it's not inflamed, the stem cells aren't sticky. In this experiment in a rat, this dark area of the brain here is filled with stem cells because this side of the brain is inflamed this brain is not inflamed, this brain is inflamed. This side of the brain received the stem cells and this side of the brain did not receive the stem cells. So all the stem cells clustered only in the brain on the side we delivered the stem cells. We can look histologically, I'm not sure how well this projects, but all these red dots here are stem cells that stuck in the inflamed side of the brain. On the other side of the brain, no stem cells because we didn't put them there. In green here are blood vessels, and in red are the stem cells that stuck to those blood vessels. The hope is that they would migrate in and, again, differentiate into the cell type of interest so that they could eventually do what we hope they're designed to do. This is how we hope to eventually move forward with a trial in multiple sclerosis, for example. These patients have lesions everywhere in their brain, optic nerve, in their spinal cord. So we can't inject every single one of those lesions surgically. We're going to have to provide another route for these stem cells, and maybe through blood vessels is the way to go. So I think I've used up my time. These are the folks at the different companies that have been involved. Of course, Doug Kerr was my mentor at Johns Hopkins, and everybody working at Johns Hopkins has been wonderful. And this is Allison, and this is how I'll accept questions. You have to raise up both hands. No, I'm kidding. Thank you.